Today we will discuss about the major macromolecules of life. In the present lecture, we will discuss about the 4 major macromolecules, their process of formation with their structure and function in detail. Macromolecules are large molecules formed by polymerization or the process of creating polymer chains out of the polymeric materials. Polymeric materials are formed when monomers are joined together by the dehydration reaction, whereas the same polymers can be broken down by the hydrolysis reactions. There are 4 fundamental types of macromolecules which are essential for the living organisms. They are carbohydrates, which are the polymers of monosaccharide units joined by the glycosidic linkage, and it contains carbon, hydrogen and oxygen. The second one is proteins, which are the polymers of amino acids joined by peptide linkages. It contains carbon, hydrogen, oxygen, nitrogen and sulfur. The third one is lipids, which are the polymers of fatty acid units joined by the ester linkages. It contains carbon, hydrogen and oxygen. And the fourth one is nucleic acids, which are the polymers of nucleotide units joined by the phosphodiester linkages, and it contains carbon, hydrogen, oxygen, nitrogen and phosphorus. Now let us discuss the given table which depicts the building blocks of macromolecules and its major functions. The building blocks of proteins, nucleic acids, carbohydrates and lipids are amino acids, nucleotide monosaccharides and fatty acids respectively. The major function of each of the macromolecules are proteins is essential for the fundamental basic structure and function of the cell. DNA, which functions as the repository hereditary information. RNA is essentially required for the protein biosynthesis. Carbohydrates help in the storage form of energy to meet the short-term demands, and lipids help in the storage form of energy to meet long-term demands and also forms the structural component of membranes. Now let us study the 4 fundamental types of macromolecules in detail. First macromolecule to be discussed is carbohydrates. They are most abundant organic molecules in nature. They are primarily composed of the elements carbon, hydrogen and oxygen. The term carbohydrate was originally coined for this class of compounds as most of them were the hydrates of carbon or could be represented by the general formula CxH2OY. To accommodate a wide variety of compounds, the carbohydrates are nowadays broadly defined as polyhydroxyaldehydes or ketones and their derivatives as the substances that yield one or more compounds on hydrolysis. Functions of carbohydrates. They are the most abundant dietary source of energy, for example 4 calories per gram for all the organisms. They are precursors for many organic compounds like fats and amino acids. Carbohydrates like glycoproteins and glycolipids participate in the structure of cell membrane and cellular functions such as cell growth, adhesion and fertilization. Carbohydrates also serve as the storage form of energy, that is glycogen, to meet the immediate energy demands of the body. Now the classification of carbohydrates. They are usually classified in 3 groups. The first one is monosaccharides or monosaccharosis, where mono stands for one and saccharon stands for sugar. The monosaccharides, often called as simple sugars, are the compounds which possess a free aldehyde, that is CHO, or ketone, that is C double bond O group, or more hydroxyl, that is OH groups. They are in fact the simplest sugars and cannot be hydrolyzed into smaller units. Their general formula is CnH2nON. The monosaccharides may be subdivided into trioses, tetroses, pentoses, hexoses, heptoses, etc. depending upon the number of carbon atoms they possess, and as aldoses or ketoses depending on whether they contain aldehyde or ketone group. Some important examples are given in table. The examples of aldose and ketose for the triose sugar are glycerose and dihydroxyacetone respectively. For tetrose sugar are erythrose and erythrulose respectively. For pentoses are ribose and ribulose. For hexose they are glucose and fructose, and for heptose they are glucoheptose and sedioheptulose respectively. The second major group is oligosaccharide or oligosaccharosis. As we all know, oligo means few, so these are the compound sugars that yield 2 or 10 molecules of the same or different monosaccharides on hydrolysis. Accordingly, an oligosaccharide yielding 2 molecules of monosaccharide on hydrolysis is designated as a disaccharide, and the one yielding 3 molecules of monosaccharide as a trisaccharide, and so on. The general formula of disaccharide is CnH2ON-1, and that of a trisaccharide is CnH2ON-2, and so on. A few examples are disaccharides like sucrose, lactose, maltose, cellobiose, trehalose, gentiobiose, mellibiose, etc. For trisaccharide it is raminose, gentianose, raffinose, rabinose, melizitose, etc. And for tetrasaccharide it is stechiose, pentasaccharide it is verbascose, etc. The third category is polysaccharides or polysaccharosis. Poly means many and saccharose means sugar. So these are also compound sugars and yield more than 10 molecules of monosaccharides on hydrolysis. This may be further classified as homopolysaccharide or heteropolysaccharide. Homopolysaccharide are the compounds which yield only a single type of monosaccharides on hydrolysis. For example, starch, glycogen, inulin, cellulose, pectin, chitin. Whereas heteropolysaccharides are the compounds which yield a mixture of few monosaccharides or their derivatives on hydrolysis. For example, hyaluronic acid, chondroitin, heparin, dermatan sulfate and keratin sulfate. Now let us discuss about the second macromolecule of life, that is proteins. They are the most abundant intracellular macromolecules with higher molecular weights and constitute over half the dry weight of most organisms. Proteins occupy a central position in the architecture and functioning of living matter. These are the polymers, that is chain-like molecules produced by joining a number of small units of amino acids called monomers. The amino acids are therefore regarded as building blocks of proteins. Functions of proteins. The first function is the structural functions. So these proteins are responsible for imparting strength to the body. It includes collagen and elastin found in the bone, vascular system and other organs. The second one is the dynamic functions. These proteins are known as acting proteins such as enzymes, hormones, blood clotting factors, immunoglobulins, membrane receptors, storage proteins that play an important role in genetic control, muscle contraction and respiration. Now let us discuss the classification of proteins. Proteins are divided on the basis of shape, composition and solubility. So the first classification to be discussed is on the basis of its shape. The first one is globular or corpuscular proteins. They possess relatively spherical or ovoid shape. They are usually soluble in water or in aqueous media containing acids, bases, salts or alcohol, and they diffuse readily. Nearly all the enzymes are globular proteins. Other examples are protein hormones, blood transport proteins, antibodies and nutrient storage proteins. The second to be discussed is the fibrous or fibrillar proteins. They possess long ribbons or fibers in shape. They are mainly of the animal origin and are insoluble in all common solvents such as water, dilute acids, alkalis and salts, and also in the organic solvents. Most fibrous proteins serve in the structural or protective role. It is a heterogeneous group and it includes the proteins of connective tissues, bones, blood vessels, skin, hair, nails, horns, hoof, wool and silk. The important examples are: number 1, collagen, that form the major constituents of the bones. The second one is elastins, that form the major constituents of ligaments and blood vessels. The third one is the keratin, that form the major constituent of skin and hair. And the fourth one is fibroin, that forms the major constituent of the silk fibers. The second to be discussed is on the basis of the composition and solubility. This is nowadays the most accepted system of classification and is based on the proposals made by the committees of British Physiological Society in 1907 and American Physiological Society in 1908. The system divides the proteins into 3 major groups based on their compositions, that is simple, conjugated and derived. So, the first one is proteamines and histones. These are the basic proteins and occur almost entirely in all the animals, mainly in the sperm cells, soluble in water, unlike most other proteins, not coagulated by heat and strongly basic in character. Proteamines are virtually devoid of sulfur and aromatic amino acids. Histones are somewhat weaker bases and are therefore insoluble in ammonium hydroxide solution, whereas the proteamines are soluble. For example, of some proteamines are clupein from herring sperm, salmine from salmon sperm, staurin from sturgeon, etc. The second one is albumins. These are widely distributed in nature, but most abundantly found in seeds, soluble in water and dilute solutions of acids, bases and salts. Can be precipitated with a saturated solution of an acid salt like diammonium sulfate or a neutral salt like sodium sulfate and can be coagulated by heat. For example, leucosin in cereals, legumulin in legumes, ovalbumin from white of egg, serum albumin from blood plasma, myosin of muscles and lactalbumin from the milk whey. The third one to be discussed is globulins. These are of 2 types, pseudoglobulins and euglobulins. Euglobulins are more widely distributed in nature than the pseudoglobulins, either soluble pseudoglobulin or insoluble euglobulins in water. It can be precipitated with half saturated solution of diammonium sulfate, can be coagulated by heat, for example, pseudoglobulin, that is pseudoglobulin from milk whey. The examples of euglobulins are serum globulin from blood plasma, ovalbumin from egg white, myosinogen from mussels. Globulins of various plant seeds are like glycinin from soybean, leguminin from peas, amandin from peach, pomelin from oranges and also tuberin from potatoes. The fourth to be discussed is glutelins. These have been widely isolated from the plant seeds, insoluble in water, dilute salt solutions and alcohol solutions, but soluble in dilute acids and alkalis and can be coagulated by heat. For example, glutenin from wheat, glutenin from corn, orizinin from rice, etc. The fifth one to be discussed is prolamins. These have also been isolated only from the plant seeds, insoluble in water and dilute salt solution, but soluble in dilute acids and alkalis and also in 60 to 80% of alcohol solution, cannot be coagulated by heat. For example, gliadin from wheat, zein from corn and hordein from oats, etc. The sixth type is scleroproteins or albuminoids. These occur mostly entirely in all the animals and are therefore commonly known as animal skeletal proteins, insoluble in water, dilute solution of acids, bases and salts and also in 60 to 80% of alcohol solutions. And they are not attacked by enzymes, example collagen of bones, elastin in ligaments, keratin in hair, tissues and fibroin of silk. The third macromolecules to be discussed is lipids. The lipids are a heterogeneous group of compounds related to fatty acids and include fats, oils, waxes and other related substances. These are oily or greasy organic substances relatively insoluble in water and considerably soluble in organic solvents like ether, chloroform and benzene. These are variously called as lipins or lipoids. The term lipid was first used by the German biochemist Fibrillar in 1943 for a major class of tissue components and foodstuffs. Chemically, the fats are defined as the esters of glycerol and fatty acids or as the triglycerides of fatty acids. The functions of lipids are: they are concentrated fuel reserve of the body, also known as triacyl glycerols. Lipids are constituents of membrane structure and regulate the membrane permeability. They are phospholipids and cholesterol. They serve as a fat soluble vitamin like A, D, E and K. They are important as cellular metabolic regulators, for example steroid hormones and prostaglandins. Now let us discuss about the classification of lipids. Fibrillar in the year 1943 had proposed the following classification of lipids based on their chemical composition. The first type is simple lipids or homolipids. These are the esters of fatty acid with various alcohols. First one is fats and oils, also known as triglycerides or triacyl glycerols. These are the esters of fatty acids with a trihydroxy alcohol, that is a glycerol. The fat is solid at ordinary room temperature, whereas an oil is liquid at the room temperature. The second one is compound lipids or heterolipids. These are esters of fatty acids with alcohol and possess an additional group also. The first one is phospholipids, also known as phosphatids. These are the compounds containing phosphoric acid, nitrogen base and other substituents in addition to fatty acids and glycerol. The second one is glycolipids, also known as cerebrosides. These are the compounds of fatty acids with carbohydrates and contain nitrogen but no phosphoric acid. The glycolipids also include certain structurally related compounds comprising the groups gangliosides, sulfolipids and sulfatids. The third one is derived lipids. These are the substances derived from simple and compound lipids by hydrolysis. These include fatty acids, alcohols, mono and diglycerides, steroids, terpenes and carotenoids. The fourth macromolecule to be discussed is nucleic acids. The most important macromolecule imparting life to all the living cell is nucleic acid. The nucleic acids like DNA and RNA are the molecular repositories for genetic information and are jointly referred to as the molecules of heredity. The structure of every protein and ultimately of cell constituent is a product of information programmed into the nucleotide sequence of a cell's nucleic acid. Although the name nucleic acids suggest their location in the nuclei of the cell, but certain of them are also present in the cytoplasm. They are the macromolecules present in most living cells either in the free state or bound to the proteins as nucleoproteins. The classification of nucleic acids. There are two kinds of nucleic acid, that is deoxyribonucleic acid and ribonucleic acid. Both the types of nucleic acids are present in all the plants and animals. Viruses also contain nucleic acid. However, unlike a plant or animal, a virus has either RNA or DNA but not the both. DNA is found mainly in the chromatin of the cell nucleus, whereas most of the RNA, that is around 90%, is present in the cell cytoplasm and a little of 10% in the nucleolus. It may be added that extracellular DNA also exists. It occurs, for example, in mitochondria or in chloroplast. Now, if we talk about RNA, then there are basically three types of RNA molecules. Number one is the transfer RNA, also known as the tRNA. The second one is the messenger RNA, also known as mRNA. The third one is the ribosomal RNA, also called as rRNA. Upon hydrolysis under different set of conditions, the 2-nucleic acid yields 3 components. The first one is phosphoric acid, a pentose sugar and a nitrogenous bases. The structural components of 2 types of nucleic acids, DNA and RNA, are discussed in the table. Now, the acid component of RNA and DNA is phosphoric acid, which is common. The pentose sugar of RNA is D-ribose, whereas of DNA it is D2-deoxyribose. The purine nitrogenous base of RNA and DNA are adenine and guanine, that is same, whereas the pyrimidine nitrogenous bases present in RNA are cytosine and uracil, whereas in DNA it is cytosine and thymine. Now, what are nucleosides? So, these nucleosides are compounds in which nitrogenous bases, that is purines and pyrimidines, are conjugated to the pentose sugar, that is ribose or deoxyribose, by a beta glycosidic linkage. What are nucleotides? So, nucleotides are the phosphoric acid esters of nucleosides. These occur either in the free form or as subunits in nucleic acids. The phosphate is esterified here with the sugar moiety. Purine and pyrimidine nucleotides fill a variety of metabolic roles. They are energy currency of the cell. In some cases, they are signaling molecules acting like hormones directly or as transducers of the information. They provide the monomers for the genetic information in the function of DNA and RNA. The 2 major functions of nucleic acids are transmission of hereditary characters, that is done by DNA, and synthesis of proteins, that is done by RNA. To summarize the importance of nucleic acid, let us discuss about the primary functions of each in brief. So, the first one, if we talk about DNA, it is the storehouse of genetic information and it controls protein synthesis in the cell. For RNA, it directs the synthesis of specific proteins. mRNA, also known as messenger RNA, it helps in taking the genetic message from RNA. tRNA, that is transfer RNA, it transfers the activated amino acid to the site of the protein synthesis. And rRNA, that is ribosomal rRNA, its functions are not clearly understood, but mostly they are present in ribosomes and are responsible for the stability of mRNA, that is messenger RNA. So, to conclude the present discussion, we have learned that all the living organisms are dependent on 4 very large molecules essentially for their biological functions. These molecules are carbohydrates, proteins, lipids, DNA and RNA. They are classified as biological macromolecules. Without DNA and RNA and proteins, all the forms of life could not exist. This is because each molecule plays an indispensable role in biology. Macromolecules constitute a major portion of the total dry weight of both eukaryotic and prokaryotic cells. They are responsible for all the most sophisticated functions of living cells, such as the catalysis of chemical reactions, the building of complex structures, the generation of movement and the transmission of hereditary information. Macromolecules thus individually fulfill many distinct roles in the cell, and in addition it works together in a large multimolecular complexes.